What if I told you that scientists can do in 3 days what can take nature millions of years to accomplish? No, this isn't a sci-fi movie trailer. It's real, and it's happening right now in labs all over the world. And it might be one of the most powerful tools humanity has ever created. It's called directed evolution, and today we're going to break it down so anyone can understand it. First, we need to clear up a common misconception. There isn't just one kind of evolution. On short timescales, generations, years, even days, life can change surprisingly fast. This is called adaptive evolution. It's how bacteria become resistant to antibiotics, how viruses evolve new variants, and how populations fine-tune existing traits to better survive their environment. On very long timescales, hundreds of thousands to millions of years, those small changes can accumulate into macroevolution. That's where entirely new forms arise. Fins becoming limbs, reptiles giving rise to birds. But macroevolution is slow, incredibly slow. The first fish appeared during the Cambrian period. About 100 million years later, the first fish with jaws finally showed up. And it took over 4 billion years for single-celled organisms to eventually evolve into, well, us. How does evolution work? It all comes down to proteins. Proteins are the tiny molecular machines that run every living thing. They're built from chains of building blocks called amino acids. And the order of those amino acids determines a protein's shape and what it does. That order is written in DNA, a kind of biological instruction manual. Occasionally, a tiny typo sneaks in when cells copy that DNA. Scientists call these single nucleotide polymorphisms, or SNPs. Most of the time, these typos don't matter. But every once in a while, one creates a protein that works better in some way. That's evolution in action. Here's a mind-blowing example. Humans and mice are very different creatures, right? But the protein produced by the FOXP2 gene, which is critical for language and communication, differs between humans and mice by only 3 amino acids. 3 tiny differences, out of hundreds, separate a human conversation from a mouse squeak. That's the power of proteins. And it's exactly what directed evolution taps into. So evolution works, but it has 2 major drawbacks. It can be extremely slow, and mutations occur randomly. Mutations happen by accident, at a rate of about 1 in a billion DNA copies. And most of those mutations either do nothing or actually break something. On top of that, even if a useful mutation does occur, nature can only work with what the environment demands. So scientists asked a bold question. What if we could speed this up and guide it towards a goal? What if, instead of waiting for nature to stumble onto the right answer, we could guide it? That's exactly what directed evolution does. Here's the basic idea. You start with an existing protein. You decide what you want it to do. Maybe it currently binds to one molecule, but you want it to bind to a different one. Then you mimic the process of natural evolution. But instead of waiting millions of years, you do it yourself in the lab, on purpose, in a matter of days. Here, step by step, is how it works. Step 1. Pick your starting protein. Scientists choose a protein that already does something related to what they need. It's your starting point. Step 2. Introduce random mutations. They introduce a small number of random changes into the DNA code of the protein. This creates a bunch of slightly different versions of the protein. Step 3. Test them. Each version is tested to see which one performs best at the desired task, which one is most fit. Step 4. Select and repeat. The best performing variant becomes the new starting point, and the whole process repeats. Amazingly, this process typically only needs about 5 rounds to arrive at a dramatically improved or entirely new protein. 5 rounds. In days. Researchers at Caltech have pointed out that even after 4 billion years of natural evolution, only a tiny fraction of all possible protein sequences has ever been explored. Directed evolution lets us venture into completely uncharted territory. Here's something even cooler. Proteins aren't just single blobs of function. They're actually modular. Think of them like Lego sets. Proteins are made up of distinct functional units called domains, and each domain does a specific job, like grabbing onto another molecule or cutting a protein in just the right place. And here's the wild part. You can mix and match these domains. Scientists can take a domain from one protein and splice it into a completely different protein, creating what's called a chimera. The resulting protein has capabilities that no naturally occurring protein has ever had. To actually produce these chimeric proteins, scientists insert the new DNA instructions into a bacterium, usually the well-known E. coli. The bacteria act like tiny protein factories, following the new instructions to manufacture the designer protein. Once the bacteria have made enough protein, scientists break them open, clean up the mixture, and end up with a vial of their brand-new, custom-built protein. The applications are enormous. In medicine, it is already being used to develop new therapies. Proteins engineered to seek out and destroy cancer cells, fight drug-resistant bacteria, or neutralize viruses. In industry, scientists are engineering proteins called enzymes that can break down plant material to produce biofuels, clean up pollution, or make chemical processes far more efficient and environmentally friendly. And here's the scale of what's possible. If you made one example of every possible sequence of even a small 100 amino acid protein, you'd produce more unique proteins than there are atoms in the entire universe. The space of possibility is almost beyond imagination. Of course, we don't need to search all of that space randomly. Functional proteins tend to cluster together, a discovery that underpins why directed evolution works as well as it does. But even within that clustered space, the possibilities for genuinely new, useful proteins are staggering. The intersection of chimeric protein design and directed evolution could, one day, give us the tools to end cancer, defeat viral and bacterial infections, and address virtually any disease caused by biology gone wrong. Evolution gave life 4 billion years to explore what proteins can do. Directed evolution hands that power to scientists with a goal, a lab bench, and a few days. We're at the very beginning of understanding what's possible, and that is pretty exciting. Thanks for watching, and please click like and subscribe if you enjoyed this video.